Right now, under your shoes, there is a graveyard holding the secrets of a 65 million-year-old planetary apocalypse.
Yeah, it's literally right beneath us.
And what's wild is that living inside that exact same graveyard is this bustling, microscopic metropolis. I mean, we're talking about a place where predators paralyze their prey to store them in underground
Oh,
vaults.
absolutely. And heavy demolition crews are just breaking apart the landscape, molecule by molecule.
It's crazy.
It is the absolute definition of an active living archive. The ground is simultaneously recording the history of the Earth, but it's also actively engineering its future.
And that duality is exactly what we're exploring today. We have a mind-bending mission for this deep dive.
We really do.
Yeah, we're drawing on two incredible documents that are just going to completely change how you view the dirt on your shoes. First, we have a fascinating educational case study on the discovery of the dinosaur extinction.
I'm right, the Alvarez case study.
Exactly. And we are pairing that with a comprehensive 2025 scientific paper from the International Journal of Modern Science and Research Technology. And that one is all about the functional architecture of soil biodiversity.
The contrast between these two sources is, well, it's just brilliant. One looks at the soil as this ancient tape recorder of planetary catastrophe, and the other looks at the soil right now, today, as a highly sophisticated living factory.
So we are going to read the dramatic history hidden in the rock, and then we're going to shrink down to explore the billions of microscopic engineers keeping that soil alive right now. Okay, let's unpack this.
Yeah, to really understand what the soil can tell us, we need to go back a bit, back to the 1970s.
Okay, setting the scene.
Right. So we're in the Italian Apennine Mountains with an American geologist named Walter Alvarez.
And reading through the case study, it's funny because he isn't out there looking for dinosaurs at all, right?
Not even a little bit.
He's studying plate tectonics. He's looking at sedimentary rocks that used to be at the bottom of the ocean to track reversals in the Earth's magnetic field.
Exactly. And you have to remember, plate tectonics was still a relatively young field back then.
Right.
But as Alvarez is out there examining these deep limestone gorges, he keeps running into this very distinct sequence in the rock.
The KT boundary.
The Cretaceous-Tertiary boundary, yes. It is a defining line of the geological record, exactly 65 million years old.
And it's super visible, isn't it?
It is. Below this boundary, the older Cretaceous rock is just packed with a huge variety of microscopic marine fossils. But then, above the boundary, in the Tertiary rock,
They're gone.
yeah, those diverse fossils practically vanish, and sandwiched right between them is this 1 centimeter thick layer of lifeless clay.
Wow, it's just a stark visual of mass extinction.
It really is. But to prove it was a sudden catastrophe, Alvarez needed to know how long it took for that specific 1 centimeter layer of clay to form.
Right, like did it slowly accumulate over millions of years as the marine environment gradually changed, or
Or did it settle out of the sky in a matter of months?
which presents a massive physics problem.
Huge. I mean, you are trying to time a geological event that happened 65 million years ago.
Yeah.
So Walter brings the problem to his father.
Luis Alvarez.
Right, Luis Alvarez, who just happens to be a Nobel Prize-winning physicist.
Handy guy to have around.
Definitely. So Luis suggests using a radioactive isotope called beryllium-10 as a timer. You see, beryllium-10 is created by cosmic rays in the upper atmosphere.
Oh, interesting.
Yeah, and it rains down on the Earth at a relatively constant rate, getting trapped in sedimentary rocks.
And because it's radioactive, it has a half-life, right? Like it decays over time at a predictable rate.
That is the exact underlying mechanism. Every 1.39 million years, exactly half of the beryllium-10 in a given sample decays into a different element.
Okay, I follow.
So, by measuring how much beryllium-10 is left in that clay layer, you can theoretically calculate how long the clay was exposed to the surface, accumulating that cosmic rain.
So it's a brilliant idea in theory. You use the decay rate as a ticking stopwatch.
Exactly.
But the case study mentions that when they actually ran the numbers, the published half-life for beryllium-10 at the time was wrong.
It was way off.
Yeah. So for the amounts they could realistically detect with their equipment, the timer in a 65-million-year-old rock had essentially run out of battery.
That's a great way to put it.
It's like trying to time a marathon with a stopwatch that died decades ago. It had halved so many times over 65 million years that the remaining signal was practically zero.
Yeah, the beryllium timer was basically useless for this specific geological time frame. The background noise would just completely drown out any usable data.
So beryllium was a total bust. Why didn't they just pack it up and study something else? I mean, go back to magnetic reversals.
Well, what's fascinating here is the nature of that pivot, because they didn't abandon the concept of a cosmic timer.
They just swapped the equipment.
Exactly, they just changed the element. Luis Alvarez suggested looking for iridium instead.
Iridium.
Right, iridium is a heavy metal that is incredibly rare in the Earth's crust. And that's because most of it sank to the planet's core during its formation. But it is much more common in meteorites and cosmic dust.
Because space dust is constantly falling on the Earth, like a very slow, invisible cosmic drizzle.
That's it exactly. So they reasoned that if the clay layer took millions of years to form, they'd find a tiny, measurable, steady accumulation of iridium mixed in.
Okay, makes sense.
But if it formed rapidly, over just a few years, they'd find almost none.
But when they actually test the clay from the Apennines, they don't find a tiny amount, and they don't find zero.
No, they don't.
They find 3 parts per billion of iridium. Now, on paper, that sounds minuscule, but that was 30 times what they expected. It was a massive, inexplicable spike.
They called it the iridium anomaly. But, the immediate challenge to that finding was localization.
Right, because a weird chemical spike in one mountain gorge in Italy.
It could just be a fluke.
Yeah, maybe a small meteorite happened to hit nearby during that era.
Exactly. So Alvarez starts digging through the geological literature to find another site with an exposed KT boundary. And he finds a cliff face in Denmark at Stevens Klint.
Okay.
They test the clay there, and the iridium anomaly is present there too.
Wow. So the soil was telling a global story. Whatever event dropped that much extraterrestrial dust on Italy also dropped it on Denmark, covering the planet.
Yeah, and that realization forced the scientific community to ask where all this iridium came from.
And the first major hypothesis they tested sounds almost apocalyptic. A supernova.
A supernova was a very compelling idea at the time. I mean, an exploding star. But to prove it, they needed to find a specific chemical fingerprint.
Right, because a supernova wouldn't just scatter iridium.
No, the intense fusion of an exploding star also creates heavy radioactive isotopes, like plutonium-244. So if the iridium came from a supernova, there had to be plutonium right alongside it in that clay.
And according to the case study, they test the clay for plutonium, and initially they find it.
They did.
They celebrate, thinking they've just proven a star exploded and killed the dinosaurs. But then, to be rigorous, they run the test again to double-check their result.
And the plutonium is completely gone.
Gone. Because it was never in the clay to begin with. Their first sample had been cross-contaminated by a completely different experiment happening in a nearby lab.
Yeah, it's a bit of a nightmare scenario for a scientist.
I can imagine. There was 0 plutonium in the KT boundary, and no plutonium meant no supernova.
If we connect this to the bigger picture, this is a beautiful, frustrating illustration of the scientific method in action.
It really is.
It is not a straight line from hypothesis to absolute truth. It is full of broken stopwatches, contaminated samples, and necessary failures. But the failure of the supernova hypothesis is what forced them to reevaluate the data.
Right. They had high iridium and 0 plutonium.
Exactly.
Here's where it gets really interesting. They pivot to the asteroid hypothesis.
Yes.
An asteroid is packed with iridium from the early solar system, but it wouldn't contain the plutonium of an exploding star, so it perfectly fits the chemical signature in the dirt.
It's a perfect match.
But I have to push back here because this is where the logic initially trips me up.
Okay, let's hear it.
How does a rock hitting the Earth kill off marine life and dinosaurs globally? Even a massive asteroid only crushes whatever is in its immediate impact zone.
That's a great question, and it's where Luis Alvarez stepped back in with the atmospheric physics. He calculated the kinetic energy of a rock roughly 6 miles wide hitting the Earth at cosmic speeds.
6 miles wide. Wow.
Yeah, huge. But it wasn't the impact itself that caused the global extinction. It was the mechanism of the aftermath. An asteroid that size would instantly vaporize upon impact, blasting millions of tons of pulverized, superheated rock and dust into the upper atmosphere.
It creates a global shroud.
Exactly.
Uh-huh.
This dust cloud would have enveloped the planet, becoming so thick that it essentially blocked out the sun.
And when you block sunlight,
Global photosynthesis grinds to a halt. The marine phytoplankton die. The terrestrial plants die.
their herbivores starve.
And then the apex predators starve. The entire global food web collapses from the bottom up.
That is just Terrifying. And I was reading in the case study that once they published this idea in 1980, it set off a global scientific hunt.
Oh, absolutely.
People started finding shocked quartz, which is quartz crystals that have been structurally deformed by massive instantaneous pressure and tsunami debris layered right into the KT boundary all over the world.
And eventually, they found the smoking gun.
The Chicxulub crater.
Right. 100 miles wide, buried under the Yucatan Peninsula. And it was actually discovered by a Mexican oil company tracking gravity anomalies underground.
The story of the KT boundary demonstrates how a single layer of clay can completely rewrite our understanding of Earth's history.
It proves the soil is a planetary archive.
But the soil isn't just an archive of the past. The chemical and physical mechanisms that preserved that iridium layer 65 million years ago are still operating right now.
Very much so.
And that brings us to the 2025 Soil Biodiversity paper. It shows that the dirt beneath us is actively being constructed and demolished by a living ecosystem. The soil is a factory building the geological layers of tomorrow.
Yeah, and the 2025 paper argues that relying on species classification for soil life is almost impossible because the sheer diversity is too vast.
There's just too much down there.
Way too much. Instead, they classify the soil metropolis by functional jobs. They break it down into 3 tiers.
Okay, let's go through them.
Let's start at the foundational layer, the chemical engineers.
This group is primarily made up of bacteria and fungi, right? And their job is biochemical transformation.
Yes. They mediate over 90% of the energy flow in the soil. To understand fungi, you have to realize they don't just sit there. They grow as hyphae, these fine multicellular threads.
Right.
In just 1 gram of soil, fungal mycelia can extend up to 200 meters.
200 meters. That's like 2 football fields of fungal threads in a single handful of dirt.
It's hard to even wrap your head around.
And the way the paper describes their function is incredibly visceral. They aren't just a generic demolition crew. Fungi actually excrete specialized enzymes directly into their environment to chemically crack open tough structural polymers.
Like the lignin in dead wood.
Right. They break down molecules that most other organisms cannot digest.
And while the fungi are actively branching out and cracking open complex polymers, the bacteria are operating on an entirely different mechanism.
Okay, how so?
Bacteria mostly live in the micropores of the soil. Those are the microscopically thin films of water surrounding individual dirt particles.
And the sheer volume of them is staggering. The paper notes that in a temperate grassland, the pure biomass of the bacteria can reach the weight of an entire cow per hectare.
Just invisible weight suspended in those water films.
It's amazing. And because they live in that liquid environment, they can pull off some wild biological tricks, like horizontal gene transfer.
Horizontal gene transfer is a fascinating survival mechanism. In those thin films of water, if a bacterium dies and its cell wall ruptures, its DNA fragments float freely in the moisture.
Just loose code floating around.
Exactly. A living bacterium nearby can physically absorb that loose genetic code and integrate it, instantly gaining new traits or metabolic abilities without needing to reproduce.
So you have these highly adaptable chemical workers absorbing DNA and processing nutrients alongside fungi cracking open wood. But this raises a mechanical question for me.
Sure.
If these microscopic chemical engineers are so incredibly efficient at breaking things down, why doesn't everything on the forest floor just decompose instantly?
That's a really good point. The answer comes down to severe environmental constraints. Remember those water-filled micropores we talked about?
Yeah.
Well, bacteria have very limited mobility. They cannot easily travel across dry soil to reach new food sources.
Oh, I see.
Because of this, more than 90% of soil bacteria at any given moment are completely dormant.
They form protective structures like endospores and just shut down.
Yes, they can wait for decades, even centuries, in that dormant state, just waiting for a rainstorm or a moving root system to bring moisture and nutrients directly to them.
So the chemical engineers are highly effective, but they are localized and often dormant. Which brings us one step up the chain, right, to the biological regulators.
Right. This functional group includes protists, arthropods like springtails, and nematodes.
Now, nematodes are wildly resilient. The paper mentions some species surviving extreme desiccation.
Oh yeah, they can handle a lot.
Literally drying out, halting their metabolism, and returning to life after 40 years dormant on a preserved glass slide. But in the soil factory, their primary job is population control.
Yes, population control is key.
Protists physically surround and engulf bacteria. Nematodes graze on fungi or hunt other nematodes. But wait, I want to push back on this ecological model.
Okay, go ahead.
If their whole job is just eating the bacteria and fungi that do all the decomposing, how does that actually help the ecosystem? Aren't they just killing the primary workforce?
That is the paradox of the trophic cascade.
Yeah.
The best way to understand it is like a financial economy.
Okay.
You see, the bacteria and fungi are incredibly efficient at absorbing essential nutrients like nitrogen and phosphorus from the organic matter they decompose.
They're good at their jobs.
Too good, almost. They are so efficient that they hoard those nutrients. The nitrogen gets locked up inside the microbial biomass.
The wealth gets concentrated and the market stalls.
Precisely. If all the nitrogen is locked inside fungal threads and bacterial cells, the plants growing in that soil can't access it.
Oh, wow.
By aggressively hunting and eating the bacteria and fungi, the biological regulators act as market correctors. They digest the microbes, take the carbon they need for energy, and excrete the excess nitrogen and phosphorus back into the soil in a mineralized form that plant roots can actually absorb.
So they are the tax collectors of the soil.
Exactly.
Without these predators hunting the microbes, the whole nutrient cycle stalls out. The plants actually rely on the microscopic predators to release the fertilizer.
It's all connected.
Okay, so we have the chemical workers hoarding the nutrients. We have the regulators redistributing them. Well, redistributing them through predation. That brings us to the top of the functional hierarchy. The ecosystem engineers.
These are the macroorganisms, earthworms, termites, ants, moles, and even the plant roots themselves.
The heavy lifters.
Right. They are the ones that physically alter the landscape on a massive scale, creating the habitats that the other two tiers rely on.
The stats on moles in this paper are terrifying if you are an earthworm.
They really are.
Moles are hyperactive excavators with astronomical metabolic demands. A mole eats 70 to 100% of its own body weight every single day, digging tunnels at a rate of 30 centimeters an hour.
And their hunting strategy perfectly illustrates ecosystem engineering. Molely saliva contains toxins that paralyze earthworms.
It's so dark.
It is. The moles gather up these paralyzed living earthworms and store them in underground vaults called larders. Some of these larders have been found containing up to 1,000 paralyzed earthworms.
1,000. That is brutal. But while the mole is doing this, it is creating massive structural changes.
Not exactly.
Aeration tunnels, mounds that mix deep mineral soil with organic surface soil. It's basically building the physical architecture of the metropolis. And we can't ignore the plant roots either.
No, we definitely can't.
They aren't just sitting passively in the dirt. They are actively engineering their environment by exuding chemicals to create the rhizosphere.
The rhizosphere is the immediate biological hotspot surrounding a root. The microbial density there is 500 times higher than in the surrounding bulk soil.
And why is that? Because the plant is running an active trade network. The plant uses photosynthesis above ground to create carbon sugars.
Yep, it's making its own currency.
Right, and it pumps those sugars down through its roots and exudes them into the soil specifically to attract and feed the bacteria. The plant pays the bacteria in carbon, and in return, the bacteria, aided by those biological regulators we just talked about, provide the plant with bioavailable nitrogen.
This raises an important question about how we view ecology. Because as neat as these three categories are, chemical, biological, and ecosystem engineers, in reality, the functional lines constantly blur.
How so?
Well, take fungi. We classify them as chemical engineers because they use enzymes to decompose lignin.
Right.
But certain arbuscular mycorrhizal fungi also secrete a highly stable, sticky protein called glomalin.
Glomalin.
Yeah. This microscopic glue binds loose soil particles together into stable aggregates, giving the soil its physical structure, preventing erosion, and creating the very micropores that bacteria live in.
So in the act of hunting for nutrients, the chemical engineer is secreting the glue that makes it an ecosystem engineer.
The ecosystem is inextricably linked. The paper stresses that this functional perspective is essential for sustainable land management.
Because if you mess up one layer, you mess up the whole factory.
Exactly. If you compact the soil with heavy machinery, you crush the micropores, suffocating the chemical engineers. If you apply broad-spectrum chemicals that wipe out the biological regulators, the nutrient economy crashes because the nitrogen stays locked up.
So what does this all mean? When I try to synthesize this, it completely shifts my perspective.
It changes everything.
The ground isn't just dirt. It is a multi-scale, interactive physical and chemical factory. The moles are building vaults for earthworms who are tunneling through ancient clay, maybe even beryllium-laced clay in the Apennines.
Yeah, entirely possible.
Right, while the roots trade carbon for nitrogen with the bacteria.
It is a perfectly balanced, invisible metropolis. And to think, we started this journey staring at a single dead layer of rock from 65 million years ago.
The history of the Earth, written in space dust and preserved by the very ecosystem we are walking on.
It's incredible.
From Walter Alvarez deducing the catastrophic end of the dinosaurs through a microscopic spike in iridium, to recognizing the bustling biological city that exists in every handful of soil today.
I highly encourage you, the next time you walk outside, just take a moment to look at the ground. You are standing on a 65 million-year-old historical archive and a living, breathing megacity all at the exact same time.
But it leaves you wondering, doesn't it? If a single layer of iridium-rich clay perfectly preserved the story of the dinosaurs' fiery end millions of years ago, what kind of chemical or biological signature are the ecosystem engineers of our current human era laying down right now?
Yeah.
When the scientists of tomorrow dig into the rock layers of the future, what story will our archive tell?
That is a fascinating question to leave it on.
Because whether it's a bustling microscopic metropolis or a dusty library, well, eventually it all ends up in the ground.